So I wanted to talk about something a little different today. It branches off of the ideas of standard assembly and parts, and I was supposed to be the chair, but I went to the wrong room and led you all to the wrong room. So I didn't get to give my intro about basic parts and explain them before Jason Kelly talks, so he'll throw me out of whack if I don't present it, so I'm going to do it anyway. All right. No, we're not working. I'll just stand here. All right, so the basic concept of a part. Parts are defined as DNA sequences encoding primitive elements of biological functions, such as protein coding sequences, promoter sequences, and ribosome binding sites. So there's several objects on this page. Which one is the part? Is it the protein, the DNA that's encoding the protein, or the sequence that is within the DNA? Now, actually, I think the community is not entirely unified in the answer to this question, but I think Tom and I are both unified in regards to believing that it's this thing, and this is an abstract representation of this thing. It's a DNA sequence. This is a physical instance of that sequence, and this is just what it encodes. But that's not necessarily something we have to agree on at this point, but in terms of understanding how we're going to define device, we need to understand that that's what we're describing when we say part. Devices then are a collection of parts that encode a human-defined function. So this is an example of a receiver device. I think it might actually be one of the compositions from Ron Weiss's work. But you can also have, in addition to these transcriptional circuit types of devices, you can have things that are biosynthetic devices. So this is the Kiesling Labs artemisinin biosynthetic device. And you'll notice it's actually 3 different DNAs, all encoded on 3 different plasmids. And that's a property of the definition of a device, is that they don't have to be on one DNA. It's a collection of parts. It doesn't necessarily imply that they're physically connected. All right, so then you have this other item, which is basic parts and composite parts. And this is kind of the BioBrick concept, where you have physical instances of the parts, which are sequences, and you have a standard assembly reaction that takes them to a composite part. And the composite part might encode a device, it might not. But in general, this is kind of the framework that we're working off of. Now, standard biological parts, then, are parts that conform to a particular set of rules, an assembly standard that is defined to allow their physical joining into a contiguous DNA molecule. And finally, then, we have BioBrick parts, which specifically mean standard biological parts that conform to a particular standard, which is the BioBrick assembly standard. All right, so I just wanted to figure out what these terms mean, but really what I want to talk to you today is about what happens when we have this wonderful technology of automated assembly, which I'm hoping we're all going to have a year from now. And we won't be talking about just, it in a hypothetical sense. We'll actually have the technology in hand and be assembling parts, really cheap, lots of them, all the time. And what do you do with it? Now, already when you do this sort of BioBrick approach to synthetic biology, you have this kind of cookie-cutter framework where you're making these basic parts, and it's very formulaic how you make the basic parts. We do this routinely with undergraduates in the lab. And I've never met an undergraduate who can't figure out basic parts in 2 days. It's really straightforward. Making composite part is technically a little bit harder than the basic parts, but still it's conceptually trivial. And finally, you make these things and then you run assays, it doesn't work, and you redesign. So the question is, why is synthetic biology so slow? Why does it take us so long to get to the desired product? Now, part of it is each one of these steps in the process is a decently long event. Making a basic part is at least a 3-day event, no matter how you do it, because there's growing of colonies, there's sequencing, these things take some time. Making the composite part in practice is what is the major time eater for us. Running assays depends on the project in particular, of course, as to how long that takes. And design is really a very minor part of what we do on a day-to-day basis. So, fundamentally, if you're going to knock out a single one of these, make it a little faster, it's going to be making the composite parts. But actually, I think none of that is really the problem in synthetic biology. The thing that really makes it so slow is that we go around this cycle too many times. So the question is, how could you exploit this new technology that's coming, right, of automated assembly to do this all more efficiently? All right, and recognizing that that's our goal, we got to look at the different aspects of design, because design is not a singular thing. First of all, like when most synthetic biology talks you go to are talking about composition design. They're talking about what the things are made from and a product study about what they made and what it does once they make it. And then you have a few talks where you hear about technical design, how you build the DNA. So that's what Jason was telling us about. That's what the Venter Institute is doing. That is what the B. Subtilis work is about. The one that we never seem to talk about is experimental design. That is, how do you design the project such that you're designing substructures or variants that all have functionality that can be measured? And I think this is an important part of how you efficiently go about getting to the desired products in synthetic biology. And I'm going to give you an example of a study we did this summer in making something called a P1 phage-mid device to illustrate the concept of a bootstrapping set and how it can speed things up. So basically what this device is supposed to do is you turn it on. It's going to be some pops in, pops out kind of thing. And it's going to turn on, and it's going to package the DNA that's in the cell into a phage particle and lyse the cell. Why you want these things, I'm not going to get into today, but I'm convinced they're useful. All right, so in general, you want to make this black box device that is the phage-mid device that has an input that is pops in and has an output that are particles in cis. In cis meaning they package the DNA that encodes the device. All right, so you go to the literature and you look to see what kind of things can you start with, and you find two sorts of things in this area. First of all, someone has made P1 phage-mids before, and what they did is they put a protein called RepL along with a site called the PACT site onto a plasmid, and they put this into a temperature-sensitive lysogen, and it packaged. Now, they don't give you any sequence data about it, so you don't know exactly what they made, but you have some idea that there are kind of minimally two functions that you need. You also have tons of molecular biology about P1 phage that tell you about what happens if you knock out a given gene or overexpress a given gene. So this is kind of the input that you have going into it, as well as you have easily can get access to the DNAs of these things. So what do you do with that information to turn that into a P1 phage-mid device? All right, so first of all, this is the way I traditionally would have done this. I would just go in, make my best guesses to what the thing should be, make the parts, assemble composite parts, and test them. So I started out and made these two things. This was about a year ago. First one, I couldn't even make it. It just killed the cells. So that one never came into existence. The second one there, I made it, but it doesn't do anything. Absolutely nothing. So this is the problem that I see a lot in synthetic biology. As you do it, you make your first shot, and you learn absolutely nothing about what went wrong based on the experiment you just did. And the question is, how do we approach the problem differently such that there isn't this wasted effort and this attempt to divine what happened wrong in the experiment and work from it? So here's the idea of the bootstrapping set. You want to ask several questions all at the same time. First of all, you want to know, are any of these proteins toxic? How toxic? That is a very common failure in the making of devices, that you have a protein that's toxic and you didn't regulate it properly. Do these genes express, or are they just duds? What composition is going to result in the desired level of expression? Do substructures of the desired device function? Do entire devices function? I think if we did this long enough, we could make a fairly long list of questions that we would want to answer right at the get-go about the design. Okay, so how do you implement this in process? Step one is you identify all the experimentally measurable substructures of the device. So in the case of the P1 phage-mid, there's only one I could come up with, which was whether they lyse the cells or not. That is something simpler than the whole thing. It doesn't have to functionally make a particle, just does it lyse? Does it turn on and kill it? You design all the parts you would expect to be a sufficient set of primitive elements for the target device, and you make them. And then you do part characterization, target device construction and characterization, and substructure construction and characterization in parallel. So all this is going to assume that you have this nice high-throughput automated assembly technology that allows you to make 96 things if you want 96 things. And certainly assaying things 96 things at a time has never been an issue. For most assays, that's pretty trivial to set up. Okay, and finally, you're also going to want to perform multiple arbitrary variants of each, and really it's just a calculation of how much money you have, what sort of access and throughput you have in terms of running a robot, as to how many variants of each thing you would want to make in parallel. So the set in this case would end up looking maybe something like this. So at part level characterization, the sorts of things that I know are an issue and the sort of things I could think are easy to look at would be expression tests, toxicity tests. If you're talking about promoter elements or ribosome binding site type elements, you'd want to do transcription tests or translation tests, something like that. You're going to have full device characterization, so maybe you want to make a few stabs at just the end goal that you're trying to make. Just make a few of them, your best guesses. And then in this case, lytic induction is the substructure device that we need for this system. All right, so here are some examples of real data of doing this. Looking at toxicity, this is a very easy thing to do. You just take all the parts, stick them into the same PBAD vector, stick them into the same cell line, put them in a fluorescent plate reader, and let them go for 6 hours measuring their OD. Follow it. You can extract out a rate constant from this. And then the rate constants in this case look something like that. So you can immediately see two things. First of all, RFP and PAC-A are about the same level of toxicity within error, so you don't really have to worry of anything about PAC-A. You could have it be constitutive if you want in your system. It just doesn't seem to have any effect on the cells. COI is kind of universally has a small effect on growth, regardless of whether it's induced. What that means, I don't know, but empirically that tells you it's not really going to matter whether you turn it on or not in your system. It's going to have a little bit of effect either way. But RepL shows this very clear effect where you turn it on and the cells stop growing. So that's clearly telling you there's no value in your next cycle of making things to having a part that has RepL constitutively expressed. But that's the sort of design information you miss if you just make one thing. I didn't actually do this experiment, but in the future we will. We just hadn't kind of done this in the lab yet. But you could also easily assay expression levels for all the basic things you want to make using various technologies, but the one that we've been looking at is STAG. The way STAG works is you put this little peptide, it's like a his tag or a MIC tag, it's just another tag. You fuse that to the end of your protein, and then there's this nice one-pot whole cell assay that's readily amenable to 96-well format that you can do to read out the expression level of your protein. So you could imagine a scenario where you do many of these. You both put STAG here and on the N-terminus, and maybe also you put different ribosome binding sites on this and look at the expression levels, and you can get a lot of information. Or if we had a well-established theory as to measurement standards, maybe this is the point in the experiment where for the new proteins you do the measurement standard experiment. All right, and then you also do substructures. So in this case, the substructure analysis, it's a very simple substructure. It's just directly turn on COI. And if it lyses the cells, great. If it doesn't lyse the cells, then you know there's something wrong with it, right? And in this case, it did lyse the cells, so great. All right, and finally, you're going to do target devices. So Will Deloach, who was a summer student, made these 5 different devices shown here. They all really are kind of the same thing. There's nothing dramatically different. They're all PBAD-induced systems. Some of them have a real PAC-A. Some of them have just a partial PAC-A, which is the packaging site itself that overlaps the gene for PAC-A. And some of them work. Some of them don't. So there's really not much information here that you can extract out as to why some of them don't work. But we got lucky in this case. We got to the end goal on the first shot just by making 5 different things. But let's say in the next round of assembly, you want to make a different composition for some reason. Maybe, for example, you don't want PAC-A to be included within the device. There's a lot of information here about general composition that you can use to make future devices. So here are the sort of conclusions you can make from this one experiment that you've done in one round of this whole process. So you know that all 3 coding sequences express at different at levels sufficient for the function of the device on the parts as you've made them. COI expression levels do not need to be tuned to get the switching responsive behavior that you want. So this is the sort of scenario where you very often have to do ribosome binding site tuning to make it be not constitutively on or constitutively off. In this case, it doesn't seem to need that. RepL is toxic, and you're always going to have to regulate it in any device you make with it. CoI and PAC-A could be constitutively expressed, though, because they have no toxic effect on the cell. And the P1 PAC site shows no evidence of even being functional, so you probably never want to use it again. So there's a lot of information that you got out of one experiment. All right, so we wanted to do this over the summer for iGEM. And yeah, I'm not going to get into their project because it's not important, but it was fairly involved, and it involved about 100 different basic parts, 90 of which were brand new, 10 of which were reused. And I should say, there was no problem making 90 new parts. It took about 1 week for them to do it. For 75%, and some of them required, a second round of picking another clone, and that took about 2 weeks. But after about 2 weeks in the lab, they were done with basic parts. Not a big deal. The composite parts is where things all kind of fall apart. So there were 932 distinct goal parts within the set. The longest assembly of those was 14 parts, 5 rounds, which in theory would take 10 days. In practice, they're still making parts. And we had this great idea we were going to do this human assembly line thing in the lab, and it resulted in a bunch of tube switching, suffice it to say. So really, the problem is this. The problem with getting to this point where we can effectively use this bootstrapping concept and speed things up is that we're still working one tube at a time. We're manually making each little thing one at a time. We're storing them in these hand-labeled boxes in the freezer. We're running them on gels one at a time. And that's not going to work moving forward. We need to get to a mode where we're automated, where everything is being programmed into a robot and done automatically and consistently every time, either in your own labs or at some sort of facility, a fab-type facility. All right, so finally I'll thank Will Deloach is the guy that did the work I talked about today, but these are all the folks in my lab, and I thank Sim Burke and Amgen for funding, and all of you for not going to see Sydney. I'll take your questions.
Your experimental approach sort of looks a little bit like what you would see in a company where you're designing experiments more traditionally, where there's a statistical approach to varying the parameters, and then you can use algorithms to extract the information out of that. Have you looked at applying some of those tools to the part level construction where you could almost, automate which set of final designs you're putting into this mix?
So I think, in terms of applying statistics requires you generate enough volume to make statistics for, but I think that's where we're heading. I think a key aspect in my mind of developing standards as a community is establishing statistics that say one method of standardization gives you a better mode of predictability than another mode of standardization. And that also applies to sets. Like for example, if you have correlations between the toxicity of a part and the failure of some type of composition, you have statistical meanings to that, you can start to interpret this data, like, how bad is the toxicity and what you should do about that in terms of design in future rounds. So I think once we have the automation and we're really doing 1,000-part sets, then you start to get a lot of data and you can really apply those tools. Right now, we literally have 30 parts. All right, thank you.